Their findings were published in Nature Communications. In 2006, Japanese scientists inserted 4 embryonic genes into skin cells and successfully reprogrammed those skin cells to act like embryonic stem cells. Artificial embryonic stem cells made from skin cells are identical to natural stem cells that develop at the earliest stages of the embryonic development process and are responsible for the development of all the cells of a fetus. However, they cannot create extra-embryonic tissues, such as the placenta. In 2015, Professor Buganim and his team were first to discover how to create artificial placental stem cells from skin cells. In this current study, the WHO research team, which included PhD students Mohammad Jaber, Ahmed Radwan and Netanel Loifer, closely examined the process that skin cells undergo to transform themselves into either embryonic or placental stem cells. We analyzed the changes that skin cells undergo to change their identity and become one of the 2 earliest types of stem cells. We looked at changes in gene expression of the skin cell, in the accessibility and activity of the DNA within the nucleus of the changing skin cell, and in epigenetic markers, i.e. marks that decorate the DNA and responsible for gene expression. These are all critical when trying to convert a skin cell into an artificial embryonic or placental stem cell, Buganim explained. The researchers found that the changes that take place in skin cells to become either embryonic or placental artificial stem cells were entirely different from one another at every level, despite the fact that both started out as skin cells. On the other hand, when those same cells were transforming into an artificial placental stem cell, the DNA sites began reorganizing themselves to allow the changing cell to implant itself and attract blood vessels, a phenomenon that occurs naturally, allowing the embryo to implant into the uterus. The most remarkable discovery came when the team compared the 2 processes side by side and looked at a chemical molecule called methyl, which interacts with specific areas of the DNA and is responsible for silencing their expression. We discovered that artificial placental stem cells contained close to 14,000 DNA sites with methyl but were nowhere to be seen in the artificial embryonic stem cells, shared Buganim. When the research team tried to understand the significance of those DNA areas, they found that they are responsible to create all the organs and cells in developing embryos, from the brain, heart, liver and kidneys to the skeleton, spinal cord and connective tissues. Make a difference. Sponsored opportunity story source. Materials provided by the Hebrew University of Jerusalem. Note: content may be edited for style and length. 2022, July 14. Scientists find primitive blueprint for embryo cell creation. Science Daily. Retrieved July 14, 2022 from ift.tt/pwli1nt The Hebrew University of Jerusalem. Thanks for watching. Please subscribe my channel for more videos.